This is going to be a little off the wall by the standards of talks that you hear at this conference, but, well, Bernie doesn't seem to mind that because he keeps booking me back. I've been coming to this conference since the beginning, and I'm delighted to be able to address you for a whole hour because that's how long it takes these days to do justice to the range of work that SENS Research Foundation does. The main way in which it's going to be off the wall is that I will be discussing technologies that would not normally be considered to fall under the heading of regenerative medicine or stem cells. And I hope I'll be able to explain why I want to do that and, explain why it makes sense to think in these somewhat broader terms when determining what regenerative medicine really is. Now, the title of my talk, the subtitle especially, really arises because I work on aging. I'm not in favor of it, and I'm interested in not only slowing aging down, but actually reversing it, actually taking people who are already in middle age or older and putting them back into a state that is both, in terms of physical and mental performance, similar to a young adult. So the question then is how to do that. And you can see why that would be described as regenerative medicine, but I guess you can also see how it would be described as rejuvenation, because that's what it is. The problem, of course, in terms of terminology, is that the word rejuvenation has somehow been already used in a manner that normally connotes cosmetic interventions. And I want to assure you that what I'm going to be telling you about today is not even slightly cosmetic. This will be extremely comprehensive, what I'm going to be talking about. I should also mention the word rejuvenation. It comes in the title of the journal that I edit. One of the major sponsors of this meeting, Marianne Liebert, is also the publisher of Rejuvenation Research, a journal that's been going for quite a few years now. I've under my leadership as editor-in-chief. And it is focused not actually only on regenerative medicine for aging, but very squarely on intervention for aging. There are probably half a dozen very respectable and reputable journals out there working on and focusing on the biology of aging, in other words, understanding aging better. But Rejuvenation Research is the only one that really focuses on doing something about aging. So I recommend you to have a look at that journal, maybe go over to Marianne Liebert's booth at the exhibit hall, and you'll find out more. But now, regenerative medicine for aging. Well, in order to really get going, and especially since I've got a nice chunk of time, I'm going to really start from first principles. And the reason I'm going to do that is because people have a very divergent range of opinions as to what they think aging actually is. That's something that I want to start by dispelling. I want to explain to you how you really ought to be thinking about aging. In other words, the best way to think about aging for purposes of, first of all, demystifying it. In other words, convincing yourself that you really do understand what aging is and it's not some kind of mystery. And secondly, thinking about it in a manner that helps to orient our thoughts around what we might do about it, how we might develop interventions. And I'm going to start with this. Fact is, we haven't done very well against aging over the years. Over the past 100 or 200 years, we have obviously made an enormous amount of progress in eliminating the infectious diseases that used to account for more than 1/3 of all infants before they even reach the age of 1, and used to obviously account for a lot of other people in childhood and early adulthood. In the Western world, that basically doesn't happen anymore, and even in the developing world, it's plummeting. But, of course, the progress against the diseases of old age has been very much more limited. We must start by asking the question, why is that? What's so hard about aging? And there are two answers that one might give. The answer that almost everyone would normally give is this. Aging is very complicated. You're not supposed to be able to read this slide, don't worry. The point here, obviously, is that an awful lot of things go wrong in the body when we get old, and moreover, they all go wrong at more or less the same time, and they interact with each other, they exacerbate each other, so the whole thing is really rather Gordian and hopeless. And that's the answer that most people would give. And it's true, that's a big reason why aging is so hard to tackle. But in my view, it's not the number one reason, it's only the number two reason. So what's the number one reason? Well, in order to introduce that, let me give you what I said I was going to give you a moment ago, a definition of aging. Here it is. Aging is simply a side effect of being alive in the first place. It is the accumulation of damage to the body that results as an absolutely inalienable consequence of the body's normal operation. This is not a fact of biology, this is a fact of physics, that any machine with moving parts is going to do this. It's going to accumulate damage as a side effect of its normal operation. And so that's actually a big part of what I was saying about demystifying aging. We all understand and we're comfortable with our understanding of what it means for a car to age. What I'm explaining to you is that the aging of the human body or any other living organism is, in essence, no different. There is, of course, a whole host of differences of detail, but let's start with the essence. So we can define damage here as changes to the structure and composition of the body that the body does not automatically repair as they happen, and therefore those changes accumulate. Those changes accumulate lifelong, even starting before we're born, and the body is set up to tolerate a certain amount of that damage, but only a certain amount, so eventually the body's performance starts to go downhill. That's all aging is. We can sum it up in 3 words. Metabolism, in other words, everything that the body does to keep us going from one day to the next, causes throughout life, causes damage to accumulate, and eventually in late life that damage is excessive and pathology results. That's a nice, simple definition. It's mechanistic, it's down to earth. That's what I'm going to use for today. Now, what's the problem? Why have we made such little progress against aging? Well, as I said, I think that there are 2 reasons, and I'm now going to introduce what I think is the number one and the most important reason. The reason is we think about aging wrongly. We think about aging like this. We think of it as somehow not like diseases. We think of various categories of disease, whether they be infections or congenital diseases or the chronic progressive diseases of old age, and then we think of aging as this kind of collection of non-specific, rather nebulous phenomena like sarcopenia, the loss of muscle mass, or decline of function of the immune system, that we don't like to call diseases. And that's a real problem, because if you talk to the general public like that anyway, or even to other scientists, and you say, well, this or that is not a disease, then what they hear is this or that is not, even in principle, amenable to medical intervention. And that is a real problem, because it's not true. The way that we ought to be looking at all of this classification is like this. So the columns are the same, the big black line is in a different place. In other words, column 3, the chronic progressive diseases that affect us in old age, is actually part of aging. The difference between column 3 and column 4 is purely semantic. It is purely a case that the things in column 3 are ones that we have chosen to give disease-like names to, perhaps because it's relatively straightforward to say whether someone has them or doesn't have them, whereas the ones on column 4 are a little bit more nebulous. But the fact is, they're all aging. They're all side effects of being alive in the first place. That's why they don't happen appreciably in early adulthood. So, if we think about disease in this way, we think about aging as consisting of all of this, then we actually get a couple of big wins. First of all, we get rid of this this implication that aging is not something that can be addressed with medicine, because everyone already accepts that we can address the things in column 3 with medicine, in principle. And the other big win is it tells us a bit more about the alternatives that might be available as interventions. This is the intervention that comes along if you don't do what I just said. If you continue to think of aging as not a disease and the diseases of old age as like infections and not like aging. What happens is you try to deal with them the way you would deal with infections. You hack away at the pathologies and you hope for the best and you try to eliminate the pathologies from the body the best you can. And of course it's not going to work because these are side effects of being alive in the first place, right? So If you try to do that, the only way you're going to succeed is by eliminating being alive in the first place, which would rather defeat the object. So, let's not do that. Now, I'm not the first person to point this out, by any means. For maybe a century or more, a few people have been observing that the geriatrics approach to attacking the ill health of old age isn't going to work. And they've said, let's try to intervene at an earlier stage in the chain of events. And that's where the field of gerontology came from. It basically says, let's try to clean up metabolism. Let's try to slow down the rate at which metabolism creates damage in the first place. Sounds like a great idea, and it seems like it ought to be possible, because, of course, in nature we see a large variation in the rate at which damage accumulates in different species. In fact, we see a measurable variation in the rate between different individuals of the same species. So, that seems like a good thing, a good hint that we might have a chance. But, unfortunately, the gerontology approach also has utterly failed to deliver any significant medical progress against the ill health of old age. And again, there are two main reasons. One reason is that if you start an intervention that slows down the creation of damage, and you only start it late in life, you don't have much to gain. You won't postpone by very much the age at which the damage reaches this pathogenic threshold. That's a shame. But the biggest reason why this approach has utterly failed is something that you all know very well. Metabolism is rather complicated. This, as I'm sure you all recognize, is a simplified diagram of a small subset of what we know about how metabolism actually works. And it's rather hairy. It's completely inconceivable that we would be able to somehow tweak this network of processes in such a way as to stop it from doing the thing we don't want it to do, the creation of damage, without at the same time having unintended consequences that would stop it from doing things we do need it to do. So it's just not going to happen, really. And of course, that's an understatement of the problem. The real problem is not so much that this is a simplified diagram of a small subset of what we know about how the body works, but rather that this is a simplified diagram of a small subset of what we know about how the body works, which, as you all appreciate, is utterly dwarfed by the completely astronomical amount that we don't know about how the body works. Even ignoring all the stuff that we don't even know that we don't know. So, forget it. It's not going to happen. Except that actually, it kind of has happened. There is a kind of get-out. But I'm warning you, this is another false dawn. About 20, 25 years ago, a couple of visionary groups in the US were working in the biology of aging, and they identified mutations in the nematode worm, Caenorhabditis elegans, that can very greatly postpone aging and extend longevity, healthy longevity. Fantastic. What's the catch? In what way does this not completely contradict what I just told you? Well, in fact, it was a massive surprise. So much so that the first group that discovered it was basically ignored until the second group came along and it became unignorable. It was a real seismic event in gerontology. It turned it upside down, made it a much more respected discipline in biology in general, brought a lot of smart people into the field. But here's the thing. It should not have been a surprise, because actually these mutations were mutations in genes that respond to involved in nutrient sensing. They respond to nutrient availability. And the thing is, it had already been 50 years since the discovery in the 1930s of life extension in rodents, not even worms, rodents, from calorie restriction. If you feed a rat or a mouse maybe 30 or 40% less than it would like, it'll live maybe 30 or 40% longer than otherwise. This is exactly the same phenomenon. And indeed, it was kind of already known to be the same phenomenon. The genes that were discovered were in a pathway that had already been explored for about 20 years in C. elegans. So basically, this is a special case that we do have the ability to extend longevity by a simple measure, namely calorie restriction or its genetic or indeed pharmacological emulation, even though in general metabolism is so complicated. And the reason we can is because pathways that have evolved that allow this to happen. It has proven useful for evolutionary fitness of species to be able to respond to the level of nutrients in a manner that adjusts metabolic priorities as between basically growth and reproduction, and thereby, as a kind of side effect, changes longevity. Fine. Unfortunately, there's a problem. Not only is this a gerontology approach in the sense that it only slows down the subsequent accumulation of damage and therefore is of less benefit the later you start, but also it turns out that longer-lived species benefit less from manipulation of these pathways. You can multiply the longevity of a worm by a factor of maybe 3 or 4 or 5 in this kind of way. You can multiply the factor of the longevity of a mouse or a rat by 1.4 if you're lucky. It's looking like only a few % in monkeys, and probably even less than that in human beings. And furthermore, this is actually something that one can pretty straightforwardly predict from evolutionary theory. So it may have been very good for a lot of gerontologists, but it doesn't seem to be very good for us, this discovery of 20-odd years ago. So we need more. And this is what the main bulk of this talk is going to be about, what SENS Research Foundation works on, and I'm going to be explaining why. Rather than trying to rescue the gerontology approach by this kind of shortcut that it turns out isn't going to work for you and me, we are pursuing a completely different third way, which I like to call the maintenance approach. And the maintenance approach really is no more and no less than regenerative medicine against aging. That's what it is. Rather than trying to slow down this process where metabolism creates damage, or this process where damage translates into pathology, instead, we separate those two processes from each other. We dive in the middle and periodically repair most of the damage so that even though it's being created, it doesn't reach a pathogenic level of abundance. And I believe, and I hope to be able to convince you today, that this approach is vastly more promising than either of the other two. Really, one starting point in convincing you of that is to go back to what I said about physics, about the fact that all machines with moving parts, whether animate or inanimate, do themselves damage as a side effect of their normal operation. We have a lot of success already under our belt in terms of extending the healthy and fully functioning longevity of simple man-made machines way beyond how long they were built to last. This is a 100-year-old car. It was probably built to last only 10 years, 15 at the outside. How come it's lasted so much longer? Preventative maintenance. Doing comprehensively to this car exactly what I just described as the maintenance approach for the human body. Going in and periodically removing the rust before the doors fall off, and so on. That's how we do this for simple machines, so there's every reason, circumstantially, to believe that we ought also to be able to do the same thing for the much more complicated machine that we call the human body. But, because it's so complicated, of course, the maintenance approach is also going to be a lot more complicated. The question is it tractable? Is it something that we can get our arms around at this point? And I believe it is. The big reason why back in 2000, 2001 I started putting these ideas out there is because I was able to classify, I was able to claim that we could classify the various types of damage, the many types of damage that accumulate in the body throughout life, into a very manageable number of categories. Just these 7 categories that you can see listed here. And as you can see, these categories are very broad, but they are also very concrete. They are straightforward, down-to-earth biological phenomena. So when I talk about cell loss here, I'm saying, simply cells dying and not being automatically replaced by the division and differentiation of other cells. That's all it is. And of course, this does happen. If it happens, then progressively the organ that is affected has fewer and fewer cells, and eventually it can't do its job. So to take a nice simple example, in Parkinson's disease, as you all know, dopaminergic neurons die at a rate much greater than the rate at which neurons in most parts of the brain die. And they aren't replaced. And sure enough, after 80 years of life, most of us have lost maybe 20% of our dopaminergic neurons. That's okay. We're still fine. but some of us lose those neurons faster than others, and those of us unlucky enough to have lost maybe 70 or 80% of our dopaminergic neurons by the age of, let's say, 80, are people who have Parkinson's disease. That's what it's all about. So that's one thing about this classification, and I'm going to go into some of the projects that SENS Research Foundation is working on by way of illustration of these, so I'm not going to spend any further time on this slide with regard to the left-hand column, but I will spend a moment with regard to the right-hand column, because the point of this classification is not simply that it's a manageable number of categories. If you take 1,000 things, you can group them into 7 different categories, however you like. That's straightforward. The question is, what's a useful classification? And the reason that this classification on the left is useful is because for each category, we can describe a generic therapy, a way to actually implement the maintenance approach that, with, of course, differences of detail, but only detail, will work for all examples within the category. So, of course, in this meeting, I don't need to explain to anybody that the maintenance approach to repair damage of the cell loss form, the top one, is precisely stem cell therapy. To put cells in that will divide and differentiate to replace the cells that the body is not replacing on its own. And the same applies right down the list. We have generic therapies. In general, therapies which are considerably less far along in preclinical and clinical development than stem cell therapies are, but nevertheless, therapies that we can describe in a great deal of detail and which obviously SENS Research Foundation is working to take forward and to progressively more compelling proofs of concept so that they will eventually be at the point where stem cell therapy is today and, of course, progress beyond that. So now, before I go to the particular projects that SENS Research Foundation is working on, I want to spend a few minutes just ramming home one really important aspect of this whole paradigm that I'm describing to you today, namely the relationship between damage and pathology. So far, I've basically, asked you to take on faith that damage causes pathology, that there are lifelong accumulations of various types of molecular and cellular change in the body which eventually contribute to age-related ill health. Now, in a way, I don't really need to go any further because, the body is a machine. Its function is determined by its structure, because that's the case for all machines. Therefore, we're done. But it would, I know, be a great deal more comforting if you had a good sense of what that relationship comes down to in practice. Some clear examples of how damage translates into pathology. So I'm going to do that for a couple of minutes. In some cases, this is straightforward. So one of our categories is division-obsessed cells, which means simply cells dividing when they're not supposed to. Simple as that. Cells losing the standard cell cycle control mechanisms. And that is more or less synonymous with cancer. So that's a straightforward one-to-one relationship. But in most cases I would say, it's much more complicated than that. If we look at the heart, we know that the heart goes wrong in a whole bunch of different ways during old age. And it turns out that those particular conditions are the consequence of different types of damage. So if we take atherosclerosis, the number one killer in the Western world, of course, then it's basically caused by molecular waste products inside cells. In other words, stuff that the cell makes and then cannot either destroy or excrete. In particular, in this case, we're talking about macrophages in the wall of the major artery, in the arterial intima, which accumulate oxidized cholesterol of various forms that it does not have the enzymatic arsenal to metabolize, and the macrophage gets poisoned by this, and bad things happen thereafter. I'm going to be talking about that at some greater length shortly because we've made some great progress in that area. That's atherosclerosis, but then there's arteriosclerosis, the hardening of the arteries, the stiffening that leads to high blood pressure in the elderly. That's caused by this thing down here, stiffening of the extracellular matrix, the accumulation of randomly located chemical bonds, cross-links between proteins of the extracellular matrix, typically caused by non-enzymatic glycosylation, by the reaction between those proteins and circulating sugar. Then we've got amyloidosis. It was, over the past several years, it's become evident that the number one killer of extremely old people, of people over the age of 105 or so, is cardiac amyloidosis. The accumulation of an amyloid not composed of the same protein as amyloid in the brain of Alzheimer's patients, but still amyloid, in the heart, which weakens it, which simply impairs the ability of the heart to beat. So, that's molecular garbage outside the cell, of course. It's, as with amyloid in the brain, it's an aggregate that forms in the extracellular space. Then finally, there's cell loss. Pacemaker cells, the cells which transmit the signal from the brain to the heart that tells the heart to beat, those are a class of cells which, just like dopaminergic neurons, do not maintain their number during life. And eventually we've got maybe only 10% as many of those as we had when we were young, and eventually the heart can stop as a result, simply because it's not getting the signal to beat, even if there's nothing else significantly wrong with the heart. So, of course, that's an example of cell loss. Now, the thing I want to point out on this slide is that everything I've told you about this is established. None of this is my hypotheses, none of this is even controversial. Anyone who works on any of these diseases will absolutely echo what I've just told you about how these diseases originate and how they progress. So, this is very good news. A solid, uncontroversial foundation for a particular range of phenomena is what you need to start with in developing, obviously, an idea for an intervention. So, this is good news. Same is true elsewhere in the body, of course. Just as an illustration, let me momentarily talk about Alzheimer's disease. Alzheimer's, of course, was defined as the combination of these things, tangles and plaques, and for a long time now we've known that it's also characterized by a lot of cell loss and synaptic atrophy. So, again, uncontroversial, completely established. And another thing, before I go on to our own project, is to come back to this business of the difference between age-related diseases and aging itself being just a semantic difference. The stuff that we don't call age-related disease, the stuff in column 4 of that table from earlier on, is just the same. If we think about, for example, decline of the immune system, immunosenescence, it's established that that is a combination of, number one, the cell loss in the thymus atrophying throughout early adulthood and eventually basically not being able to make any more T cells, which eventually comes back and starts to hit us late in life because our T cell compartment doesn't have enough versatility to cope with infections. And secondly, having too many cells. This thing here, having too many cells because cells are not dying when they're supposed to. Turns out that cytotoxic memory cells, memory T cells in particular, actually get into a state when they've been activated too often by persistent viruses such as cytomegalovirus. They get into a state that very much resembles replicative senescence. The cells no longer proliferate in response to antigen, but they do also refuse to die. They refuse to respond to apoptotic signals. So, it's well established that these are the drivers of immune senescence. Okay, so I think I've probably said enough to generally get home the point that this is a good way to think about aging from an intervention perspective. But now I want to move on a little bit and talk about specific projects. Because first of all, I want to give you a feel for how tractable this is, why it's reasonable for us to be optimistic at this point about the rate of progress and the possibility that these therapies will come along and actually reach the clinic in the next 10 or 20 years. And secondly, I want to emphasize, perhaps above all, the fact that we are talking here about combination therapies. I'm sure that all of you in your various roles in the stem cell field are aware of the limitations that researchers have been encountering with regard to the ability of stem cells to do their job. And you are, I'm sure, pretty much all familiar with the fact that one of the big limitations is that when stem cell therapies are applied to elderly people, they don't work nearly so well as in the young, even if we're using exactly the same protocol with cells from exactly the same source. Essentially, that stem cells have the ability of stem cells to do whatever job we'd like them to do depends not only on their intrinsic properties and genetic and epigenetic potential, but also on the environment in which they find themselves after they have been introduced, after they've been transplanted. And that's why, to my mind, it's absolutely clear already that stem cell therapies will reach their full potential only once other therapies, if you like, adjunct therapies, of the sort that I'm going to be summarizing in a moment, are also available to be co-administered, especially in the case of applying these things to the diseases and disabilities of old age. So, first of all, let me talk about a very straightforward case, something that's very much within the conventional regenerative medicine world. We've already heard today, for those of you who were in Doris Taylor's talk this morning, about the concept of decellularization, taking an organ and bathing it in a nasty cocktail of acids that essentially eliminates all of the cells but leaves the extracellular matrix intact and therefore leaves the vasculature intact. This is a cool way of sidestepping the difficulty of actually building a vasculature, because then you can simply reseed this scaffold with cells of your chosen type, of your chosen origin. For example, you can make an autologous organ just by getting cells from the prospective patient, and this really does seem to work because the extracellular matrix itself is only very modestly immunogenic. We are not trying to duplicate other people's efforts in this area. By and large, we don't do much stem cell therapy or tissue engineering simply because so much of it is already being done by folks like you. But we did identify a bit of a gap, a bit of an area that was very important but not being significantly worked on a couple of years ago, and that's this. I already mentioned that the decline in function of the immune system in old age is partly driven by the shrinkage of the thymus. We're building a new thymus, and we're doing it at Tony Atala's institute, the Wake Forest Institute of Regenerative Medicine. Tony has been a great supporter of the paradigm that I'm describing to you today for many years, and a great colleague, and this is one of two projects that we're doing at that institute right now. The other one I'll come to in a second, because it's part of what we're doing about cancer. We believe that cancer is by far the hardest aspect of aging to fix. The reason for that is straightforward. It's the one that has natural selection on its side. It gets more and more ingenious the more ingenious we get, because that's what selection is all about. And we are focusing on telomere control. We believe that this is the one universal Achilles heel of cancer, that they always have to find some way to compensate for the telomere shortening that occurs during DNA replication. And we feel that the groups that are already working on this are going along the right lines, but we feel that we need to be more aggressive than they're being. In particular, Geron, a company with, of whom there are many friends in this conference, has for some time now been working on an anti-cancer therapy based on telomerase inhibition. We think that's great, but we think that, first of all, we can't only focus on telomerase. We've also got to focus on ALT, the telomerase-independent mechanism of telomere elongation that about 15% of human cancers use. And we have a project at our research center in Mountain View working on that. But also, we feel that there's a big problem with cancer, namely that at the moment, we have to, all the therapies that we're trying of every kind work by being selective, by going for some feature that distinguishes cancer cells from non-cancer cells. And the big difficulty, of course, is that cancers are rather good at masquerading as non-cancers, at figuring out how to mutate into a form that gets on the wrong side of the criterion, whatever the criterion may be. So our view is that to take advantage of the fact that if we control telomere length, then no cells are actually affected for a long time until they've divided enough for their telomeres to get short enough. And in particular, that even after 10 years, only the stem cells of the most rapidly renewing tissues, such as the blood and the gut lining, are going to be affected. And we're working on exactly those things. We think that we should be able to use periodic stem cell reseeding to fix that. So at Wake Forest Institute, we're doing that in the gut. It turns out that the gut lining can be reseeded using mesenchymal stem cells and endothelial progenitor cells from the circulation, which can be encouraged in various ways to invade the gut wall from the circulation and get into the crypts. A project that we've just finished in China, looking at the blood, is something that I'm going to show you some actual data on, because we can. This was a project which specifically looked in mice at the impact on the blood of telomere shortening, and which showed how we could fix it using stem cells. So the numbers do not look particularly impressive here, simply because of the limitations of the mouse model we were using, but we're extremely pleased with the result. I'll explain why. Mice that do not have a gene for telomerase are actually fine, because mice normally have really long telomeres, and they can just get away with no telomerase for their whole life. But if you inbreed mice with a knockout for telomerase, then, of course, each generation gets born with shorter telomeres, and eventually you end up with mice being born with telomeres so short that they do suffer age-related problems as a result of telomere shortening. Now, the problems they suffer are in the gut and in the blood at about the same time, which means that doing an experiment, a lifespan experiment, just looking at the blood is rather difficult. But luckily we were able to exploit a discovery from several years ago by a group that we actually collaborated with on this project, in which there's another mutation we could cross in that delays the pathology in the gut by a substantial amount and does not significantly delay the pathology in the blood. So those mice only get anemia, basically. And that's what this red line is. These are the negative controls, which had their lives considerably shortened by telomere problems in their blood. The positive control, the green line, were those same mice, but those mice had been given a bone marrow transplant with long telomeres. So they got problems in their gut. They lived a bit longer, quite a bit longer, and then they got the same problems, but they got the problems in the gut. Now, the black line is the actual experiment. The black line is the survival curve of mice which, again, were given a bone marrow transplant to rescue their blood, but the bone marrow transplant was from mice that also did not have a telomerase gene. These were also knockouts. The only difference was that those knockouts were early generation ones which had nice long telomeres. So what this does, this experiment, is it very robustly falsifies the concern that a lot of people had with regard to this whole concept, namely that telomerase might have other functions over and above the extending of telomeres that might be critical for stem cell function. This shows, at least in the blood of mice, that that's not the case. Then we're working, as I mentioned death-resistant cells, cells that accumulate because they're not dying when they're supposed to. They've lost the ability to respond to apoptotic signals. Cells that of the classical replicative senescence phenotype that was discovered 50 years ago by Hayflick do accumulate in the body, albeit not to very large numbers, but they may be actively toxic. People are interested in getting rid of them, and so we're looking for drugs or indeed suicide gene therapy approaches to get rid of them. We're collaborating with the Buck Institute on that. And we're also, in the case of the immune system, looking to actively, physically remove these cells from the body if they won't die, because they're in the circulation, and we can do this with pheresis. In order to do this, we have to be a bit sophisticated, because the markers that distinguish these cells that we want to get rid of are not unique. So one has to look for a combination of markers. It's useful to be able to look for absence of markers, and so we're creating a rather more sophisticated system than had ever existed before to do this kind of multiplexing of pheresis, and we're doing that with a group at Berkeley. Now I'm going to show you some more data. So we're interested in molecular regenerative medicine as well. In other words, cleaning up the inside of cells and also the extracellular space. I explained earlier that a number of our different types of damage, as we classify them, are of that nature. And one of those types of damage is simply molecular waste products that accumulate inside cells. Many of you have probably heard of lipofuscin, mainly proteinaceous substance, fluorescent substance that accumulates in the heart. It's been a major marker of aging since forever. In fact, even since before the lysosome, the place that it actually resides, was discovered. We're interested in a couple of slight variations on that theme, which are the causative agents in a couple of the most important diseases of old age. First of all, macular degeneration, which is caused by the accumulation of a strange molecule called A2E that arises as a reaction product between fatty acids and vitamin A that's involved in the visual cycle. And secondly, we're looking at atherosclerosis, the number one killer, of course, in the Western world, which is mainly driven by the accumulation, as I mentioned earlier on, of oxidized cholesterol. Specifically, there's one particular oxidized cholesterol variant called 7-ketocholesterol, which most people believe is public enemy number one here in terms of abundance and toxicity. And at Rice University, we are working on exactly that. This is the nature of the problem. A picture is worth 1,000 words, as always. Macrophages, throughout life, enter the walls of our major arteries for the purpose of cleaning up detritus, essentially engulfing stuff, recycling it, and exporting it for reuse. And they're very good at it. But macrophages do not like oxidized cholesterol. It gets them in trouble. And in particular, it messes up their lysosomes. Lysosomes are an essential part of how all cells work, and certainly how macrophages do their trafficking. And when lysosomes get inactivated by 7-ketocholesterol, this is what happens. They fill up with lipid, the whole cell fills up with lipid droplets, and it's no longer called a macrophage, it's this kind of undead thing called a foam cell. And you've all got foam cells. In fact, you all had foam cells already when you were kids in your aorta and your major arteries. It's just that you didn't have very many of them. But they accumulate throughout life, and eventually there are enough of them that the surrounding vascular smooth muscle cells start to get angry, inflammation arises, and the plaque attracts more macrophages, which become part of the problem, and the plaque grows, and eventually, of course, it bursts, and you get a heart attack or a stroke. What are we doing about it? Well, this is a really quite good illustration of how different the maintenance approach is from other approaches. The geriatric medicine approach to combating this phenomenon, of course, is basically stents. Go in and ream out the fatty deposits of an advanced plaque and put in some kind of tube to stop the artery from constricting. The gerontology approach, if you like, prevention of the creation of damage, is statins. And we all know the problem with statins, that rather than attacking the oxidized cholesterol, they actually inhibit the synthesis of normal cholesterol, which, as you all know, is an essential molecule, without which we would be very dead indeed. So there's only so far you can go with statins, and as we know, they don't work very well, and they have big side effects. So this approach that we're taking is to attack the actual oxidized cholesterol. And what we're doing is we are augmenting the enzymatic arsenal of the macrophage. We are giving it an additional enzyme encoded by a gene that we found in bacteria that allows it to break down 7-ketocholesterol so that it never gets poisoned by the 7-ketocholesterol. It took us a year or so to find such a bacteria. When we started this project back in, I'm going to say 2007, 2006 maybe. It then took us another year or two to figure out the genetic basis, the genes and enzymes that the bacteria were using. And then we had the third problem, the much harder problem, which was to modify those genes so that the enzyme actually works in the human cell. Obviously, human cells and bacteria are very, very different. Not least, bacteria don't even have lysosomes. But a couple of years ago, we were able to get it working. This was the result. This was published in 2012, as you see, but we've been making progress since then as well. Essentially what this shows is simply that if you have absolutely insuperable amounts of this horrible substance, then all the cells die really quickly, whatever. But if you have more modest concentrations of 7-ketocholesterol, then the right-hand bar is taller. This is, of course, viability, and it shows that these cells are very substantially protected, that they are much more able to survive in the presence of a certain quantity of this substance than these various negative controls, cells that don't have the enzyme, or they've got the wrong enzyme, or they've got the right enzyme, but it's not targeted to the lysosome. So this is all extremely heartening. We're getting it working in macrophages. We will, of course, very shortly be trying to move on to mouse models, and we think that this is really a chance of a proper cure for We have a bunch of other projects here. I guess I'm beginning to run a little short of time, so I won't go through the first two. I'll just talk about the last one here, extracellular waste products, amyloid I mentioned in particular. So you all know about amyloid in Alzheimer's disease in the brain. In the case of the heart, as I mentioned, it's a really important characteristic, thing that kills really old people at a very high rate. It seems to be the number one killer of supercentenarians. And two labs that we're funding, one at Harvard and one at UT Houston, we are looking at ways to stimulate the immune system to deal with this. The clinical trials that happened over the past few years looking at Alzheimer's amyloid looked at a way to stimulate the immune system to engulf amyloid, essentially to stimulate the microglia in the brain to internalize the material and break it down. And the results were underwhelming in terms of cognitive benefit, but they were very good indeed in terms of the actual goal, the elimination of amyloid. So I think it's extremely good that we have that technology under our belt as something to bring back out in co-administration with future therapies that get rid of tangles and replace cells and such like. In the heart, however, we don't have these complications. The amyloid seems to be the clear actual agent that causes the problem. So if we can attack the amyloid, we expect to have clinical benefit with just that one therapy. And we are trying two different immune approaches. One, developing neutralizing antibodies that simply protect the cardiomyocyte from the amyloid, and the other one being a very curious phenomenon that we're extremely excited about, which is antibodies that don't merely bind to their cognate antigen, they actually chop it up. They're proteolytic. These were discovered by this group in Houston and have shown promise in the case of Alzheimer's amyloid, and we're using them against transthyretin amyloid. So just a couple of slides of data on these. This is the Harvard project, the neutralizing antibody, and this is just very preliminary new data, so I can't really elaborate on it very much at this point, I'm afraid. But it's simply showing that if you take a concentration of amyloid that halves the viability of cells in culture relative to cells that do not have amyloid, then you can apply our lead antibody and you entirely restore viability. You take viability straight back up to 100%. So we're extremely heartened by this. It's obviously just very early days, but nevertheless a great start. With regard to the proteolytic antibodies, which are called catabodies, the issue here is selectivity. Transthyretin, the protein in question, that is the main component, the main constituent of cardiac amyloid, is, of course, an essential protein which carries thyroid hormone around the body, and it normally acts as a tetramer. It forms a nice cage around the thyroid hormone, and that cage is not amyloidogenic. That tetramer is fine. The problem is that if a molecule of transthyretin gets bent out of shape, then not only does it no longer form the tetramer, stays as a monomer, but also it's got hydrophobic residues exposed and it forms aggregates and fibrils, and that's where amyloid of transthyretin comes from. So what we want is an antibody that binds and chops up the monomer but leaves the tetramer alone. And as you can see here, that's exactly what we have. This antibody is exposed to both of these things in the same concentration, and the monomer gets chewed up very quickly and the tetramer is completely preserved. So we're very happy. Again, very preliminary data, but not all that preliminary. We got it published in JBC a few months ago, so we're fairly happy with how this is all going. And we have a bunch of other projects that I also really don't have time to talk about that don't entirely fit into the 7-point plan. They're more context of the plan. We are very much a goal-directed organization, a humanitarian organization. We're not in this just to publish or just to get rich and famous. Not at all. We're into this to save lives. And a number of these projects are actually to do with accelerating regulatory approval of complex combination therapies like this, also preventative therapies, because a lot of this will be preventative, and getting people more comfortable about the whole thing by exploring the demographic and economic implications, and so on. So we have quite a lot going on, as you can see, and I hope I've been able to convince you that the range of things that we're doing here is a range that people who are interested in stem cell therapy should be interested in. That if you think that you want stem cell therapies to really deliver on their potential, on their full potential, especially in relation to the largest market out there, namely the elderly market, then you'd better be looking at combining those stem cell therapies with other types of regenerative medicine, let's call it molecular regenerative medicine, that will potentiate stem cells such that the two things working together will give far more benefit than either would give on its own. If you want to know a lot more, you're welcome to have a look at this book that I put together about 7 or 8 years ago now. That sounds like a long time, so you might think, well, it's rather out of date, isn't it, given all this progress? But, of course, the great news is that the progress has been very much as we predicted. The 7-point paradigm that I've been describing to you today is essentially it's basically what I put forward in the year 2000, 2001, and it has gone through pretty much no change since that time. Not because I haven't been listening, not because progress has been minimal, just the opposite. It's because progress has borne out the correctness and the validity and the promise of this paradigm. It seems to be standing the test of time, and that's got to be good news. So I'll stop there, and I'll be happy to take questions for the next 5 or 10 minutes. Thank you. Oh, yes, I mentioned this morning, but I should probably mention again, we have a booth downstairs. If you want to know more, pick up a copy of our annual report, which is about a month old, I guess. My colleagues down here, Maria and Gary, are staffing it, and our VP of Outreach, Jerry Barrett, will be arriving in a couple of hours and will be here for the rest of the conference. So there will be a lot of other people around in addition to me for you to ask questions if nothing occurs to you right now. Go ahead, please.
Just a point of personal interest that you kind of glossed over a little bit. I know.
So I don't think the microphone's actually working, so raise your voice.
Oh,
please.
Okay. I'll just talk louder then. So you sort of glossed over the mitochondrial mutations that you'd put up there.
All right.
And since we have some time, I was wondering if maybe you could say a word on what you're kind of working with that.
Sure. I'll spend a couple of minutes on this. So mitochondrial mutations are a category out of our 7 which are a bit of a Cinderella in the sense that unlike all the others, there is no major pathology of aging that we're aware of that can clearly be blamed mainly on mitochondrial mutations. They might just not matter very much in aging. They just might not matter very much. But our view is, if they might matter, we'd damn well better figure out a way to fix them. It would be a bad idea. We'd feel pretty dumb if we fixed everything else and then people carried on dying on schedule because of that one and we hadn't bothered working on it. So let's not do that. So the question is how to actually address the problem of accumulating mitochondrial mutations. And various ideas have been put out there over the years. Our favorite one is something that was first proposed in the mid-1980s, which is to complete the job that evolution has left almost, but not quite complete, namely the transfer of mitochondrial genes to the nucleus. Only 13 proteins are encoded in the human mitochondrial DNA out of well over 1,000 proteins that the mitochondrion is composed of. And the machinery that allows nucleocoded proteins to be synthesized in the cytosol and then imported into the mitochondrion is very well characterized, so it really ought to be possible to co-opt it for these additional 13 and make, if you like, backup copies, as I put it here. This is the phrase that's often used, allotopic expression. There are a couple of approaches here that look very promising. One is to actually not import the protein at all, but to import the messenger RNA and have the RNA translated in the mitochondrion. And the other is to cause the protein to be imported what's called co-translationally. In other words, to be imported while it's still being synthesized on the outside surface of the mitochondrion. This is something that a group in Paris figured out a few years ago how to control using well-chosen 3' untranslated region in the message that causes the message to be targeted to the outside surface. And both of these approaches are, to my mind, very promising approaches to overcoming what's perceived as the major problem, the major obstacle to making these backup copies work, namely the hydrophobicity of the proteins, which severely impedes the ability to the cell of the mitochondrion to import them. We're making good progress on both of these approaches. We believe we're actually, well, I wouldn't say we're close, but we're a lot closer than anyone else has ever been.
Okay, thank you.
Wonderful talk.
Thank you.
You listed there the talk about the blood stem cell receding and gut stem cell receding. Can you talk a little bit more?
Sure. Let me see if I can go back to the relevant slide. Right, so in normal aging, in humans, there isn't all that much that goes wrong with the rapidly renewing tissues, the blood and the gut lining and the epidermis. The wrinkling of the skin, for example, and other skin problems are generally problems of other cells than the epidermis. They're generally to do with thinning and stiffening of the dermis, for example. So you might think we wouldn't really have a problem to solve here. But if we do what we're planning to do with our anti-cancer approach, namely an indiscriminate assault on the ability of cells to actually extend their telomeres, and when I say indiscriminate, I mean going after all of our cells, whether or not they are cancerous, then we will be creating this problem. We will be creating a side effect where, within 10 years or so, maybe 20, stem cells of the blood and the skin and the gut will simply no longer be there because their telomeres will have become too short and they will have either died or differentiated or whatever. So this is to solve that. We want to repopulate those stem cell pools. We want to show that it's possible to repopulate those stem cell pools in a manner that allows the tissue to be perpetuated indefinitely, even though the individual stem cells do not have the immortality that they have at the moment. Now, I know, of course, that this is an extremely elaborate approach to combating cancer, and I hope to God that we're not going to need to do it. But I do not want the last 40 years of over-optimism with the war on cancer to be continued any longer. I do not think that it's yet time to be relying on, for example, various immunotherapy approaches to cancer to actually do the job that has not been done by cancer specialists so far.
Thank you so much.
Go ahead. Go to the microphone, though. I think it's working now.
You talked briefly about calorie restriction, but overall, with individual mechanisms that are approached, do you think an approach would, in terms of humans, calorie restriction and increasing the lifespan, reducing metabolic oxidation?
So what I was trying to say is that it seems, both from theoretical considerations and from the data that we have that's available, it seems that longer-lived species get very much less benefit from calorie restriction than short-lived species do. Now, that's a big problem because it means that also any of these emulations of calorie restriction that we may achieve by genetic manipulation or by pharmacological manipulation, such as rapamycin, for example, will also have the same problem because they're activating the same pathways, the same network of pathways. If you don't have a network of pathways, then you're not going to be able to activate it, right? And of course, the focus in gerontology over the past 20 years, since this was discovered, has been on the areas that are conserved, on the master control genes, if you like, that are at the apex of this pathway, that actually do the sensing and, initiate this cascade of effector pathways that actually implement the readjustment of metabolic priorities. And those genes certainly are conserved throughout evolution, even as far back as yeast. What's missing is study of the downstream genes, the downstream pathways that actually do the job, that lower oxidation, that upregulate repair, and so on. Those things seem to be the ones that are weaker, are less effective in longer-lived species. And, it's a shame, but it's how it is. So it would be a great pity if gerontology continued to be overly fixated on trying to get the most out of that approach at the expense of looking at approaches that will actually work.
So you just talked a little bit about antioxidants, and I was curious if there's any work on that in the cleanup of the metabolism.
Yeah, so I didn't really talk about antioxidants, and the main reason why is because I don't hold up much hope for them either. Of course, antioxidants have been terribly popular in the field of anti-aging medicine since forever because we actually have some, and some of them are nicely patentable, and you can make money out of them. But the unfortunate fact is they don't seem to have much benefit, and there's a good reason for that. You would expect antioxidants to have a benefit in a pathological situation where someone has a specific disease where their free radical production is through the roof for some reason or other. But if they're basically normal, if they're basically just as healthy as they can be expected to be for their age, then the problem is that free radicals are not just a bad thing. Evolution has had to make the best of free radicals ever since evolution invented breathing 2 billion years ago with the endosymbiotic event. And it has, as evolution tends to, made the best of a bad job and found uses to put free radicals too, as signaling molecules in particular, in various ways. Which means that if you were to obliterate all the free radicals from your body right now, you would not be happy at all. And evolution, of course, and our metabolism knows this. So what happens is that if one actually takes a big dose of dietary antioxidants and drives down the level of free radicals in the body, then the body responds by dialing down its own inbuilt antioxidant machinery, the enzymatic antioxidant machinery, so as to essentially compensate and basically laugh in your face and do what it was doing anyway. And this is the general belief of why antioxidants don't work. In fact, this all goes back to Denham Harman, the originator of the free radical theory of aging, one of my extreme heroes and role models, who actually died at the age of 98 just a few days ago. Harman did plenty of experiments in the 1960s trying to see what he could, how much he could extend mouse and rat longevity using antioxidants. And basically he got a good deal of defense against infection and elimination of early life mortality, but maximum lifespan was not extended at all. And like a good scientist, like the enormously awesome person he was, he carried on thinking about that. He continued to worry about it, and eventually in 1972 he published a very much less well-known paper identifying the mitochondria, and particularly the mitochondrial DNA, as potentially the explanation. Basically, his explanation, which is a little bit out of date now, but still was extremely prescient, was that antioxidants simply don't get into the mitochondria enough, so they can't stop the free radicals from doing their damage. So that's not quite true, but it's the kind of thinking that one has to, elaborate thinking that one has to engage in order to understand this whole area.
So is there any possibility of using that in the mitochondria if you were doing that in a cell culture system?
Right. So some people have taken Harman's idea forward and have tried to develop antioxidants that specifically reside in the mitochondria. In fact, the best idea for doing this was to attach a cation adduct to antioxidants that is built in a way such that the charge is extremely delocalized and therefore does not prevent the molecule from passing through the mitochondrial membrane. And this kind of works. You can get things selectively into mitochondria. But like I say, it's not only the localization that's the issue, it's also the issue that free radicals are a good thing as well as a bad thing. So benefits there have also so far been extremely limited.
Okay, thank you.
So that actually seems to be it. Thank you very much for your time.